Hello viewers, welcome yet again to another lecture on the genetics of Mendelism and beyond. In our earlier lecture, we have been talking about the various facets of sexual dimorphism, and we had left at the point where we found that in some cases there were certain disadvantages of sexual dimorphism also, not always advantageous, because if the plumage was very sharp, very attractive, very colorful, then it was an open invitation to the predators also, because they would like brighter colors or a more striking appearance. Now, picking up a thread from that discussion, let us go beyond to some of the advantages also of sexual dimorphism. We find that there is a classical example of dimorphism where it would be really advantageous in what is called as courtship of the birds. Now, this is a bird of paradise, very beautiful and brightly colored plumage containing bird, and these birds, zoologically called as Lophoria superba, they do not have natural predators in the place of their origin. So they are not bothered as to whether they are brightly colored, they are not prone to the predators. So they have a very striking plumage, they have long feathers on the tail and even head, and in spite of all these assets, they are not susceptible to predation. So in fact, this feature helps them to attract the females. And it is not only the case of attraction, but this is also a kind of their visiting card, which means the type of plumage they have, the extent to which the plumage has been developed is going to give an information about the male's state of health and the possibility of having healthy offspring. So that is also of a great advantage. In case of plants, if we take an example of Saponaria officinalis, which is also nicknamed as bouncing bet, we find that the sexual dimorphism of the staminate and the pistillate flowers is highlighting the pollination mechanism by the pollinators. They have totally distinct morphology and which is congruent to the mouth parts of the pollinators. Coming to the sexual dimorphism in humans, we know it's a matter of common sense that at puberty we would find that women and men they would begin to develop their sexual organs and they would be definitely visually different from each other. Hair would appear on the armpits, on the pubis, face, legs and other parts of the body, and genetically it is seen that men have more hair on the face and other areas of the body. And then which is very interesting, that both men and women also have hair on their upper lip. And of course, the women present in the development of mammary glands would be a unique feature, and this is also regulated by genetics and hormones, although not all of them would have the same degree of development. So that means when we now discuss what are the actual causes of this sexual dimorphism in case of animals, what could be the possible mechanisms or the possible reasons? The first which comes to us is genetics, because we know that the main cause of sexual dimorphism would reside in the sex chromosomes. We will ask ourselves a question later: is it only the sex chromosomes which are going to decide the sex of an individual? Perhaps not. So usually the females would have two X chromosomes and the male would have one X and one Y. Now this is only one type, just one type of the sex determining mechanism, as we would see subsequently. There are other ones also. In some cases, for example, if you are looking at some invertebrate species, the females would have one sex chromosome and the males would possess two. So that means there would be one chromosome less than the other counterpart. So that means genetics plays a very, very vital role in deciding the sex of an individual. Number two, the hormones are also very important and vital, because you know that each sex is differentiated by different concentrations of hormones. The hormones in male and female are distinct, and it is during embryonic development of certain species, if there is a high concentration of testosterone in the brain, this would cause them to develop as a female. Otherwise, they will develop into a male. The third factor which plays a very important role is food itself, because it's a key factor. As you know, food is needed for the proper development of the secondary sexual characters, which would keep in place the property of dimorphism. How would you imagine that a sick and a poorly fed animal will exhibit good dimorphism? So that means in that case he or she will not be able to attract the opposite sex at all. So food is also a vital factor. Of course, it is not the only factor. After genetics and hormones, it is an important one. And very interestingly, in certain animals, the season of the year is also very important, because what is the actual mating season, which would then cause the dimorphism to appear in certain species, because in the rest of the year they will not show sexual dimorphism, and it will only be during that mating season that they will show distinctions. This type of phenomenon is highlighted in an example of Hawaii akepa, which is Loxops coccineus. If you just see on the screens the two birds of Apici, you will find that usually on the right side of the screen, all the birds, they are brownish. And at the time of the mating season, the males, they develop a very attractive orangish type of hue in their plumage, in their feathers, which makes them attractive and even distinguishes them as a male, and this is important for mating. So that means now we can conclude that the phenomenon of sexual dimorphism has been in fact a biological riddle for thinkers and biologists of all time, because they have been wondering as to what really causes this type of distinction, and people have always tried to know the factors which determine the male and female sexes of a particular species. But then when we come over to the mechanisms of sex determination, we have to then talk about four very important aspects. On your screens you would find the four categories that we have to discuss subsequently. One of them is genetically controlled mechanisms, where we will talk about the genes and the chromosomes which are deciding the sex of an individual. The second would be metabolically controlled mechanisms. Are there certain factors which are, when blocked or when activated, may give rise to a change or a reversal or an alteration in sex of an individual? It is possible. And number three is the hormonal control mechanisms, which means what is the relative proportion of the hormones which is going to decide as to what is going to be the future sex of an individual. And last but not the least It is the environmentally controlled mechanisms. Now, we should not think that the mechanism of sex determination is entirely based on genes, because as we would see after some time, the environmentally controlled sex determination is a very, very important and a primitive means of sex determination, and it has then gradually evolved into the genetic ones. So the genetically controlled sex determining mechanism would take into account the first part, and that is the sex chromosome mechanisms, and this phenomenon is called as heterogenesis, because we would find that the two chromosomes, namely X and Y, they are distinct. So there has to be a balance, and this would lead us to a discussion of genic balance mechanism as to, is there a proportion between the X chromosomes and the autosomes? And then in some cases there can be male haploidy, which means if the constitution is haploid, then it is a male, and if it is a diploid, then it becomes a female. And there are other terms like haplodiploidy. And then finally there are certain single gene effects also. First, let us talk about the genetic basis of sex determination, and we find that in most of the organisms the genetic control is the one which is important, and then if it is genetic control, then the environmental factors are not going to be much operative here. And we would find that there are certain genes on the chromosomes of the higher organisms. They are going to regulate the determination of sex, and at the time of fertilization the sex of an organism is determined. So we can distinguish, as you see on your screens, two basic genetic mechanisms of sex determination, which is genic sex determination and the genotypic sex determination. We generally talk about genotypic sex determination because we are always obsessed with X and the Y chromosomes. But then there are other types of sex determination also where sex chromosomes are not involved, and this is called as the genic sex determination. And this is a very classical example of fishes. In most of the fishes we find that there are independent genes which are located on different chromosomes, and they are the ones which are responsible for sex determination. For example, if we take a hypothetical example, there is a male which has the genotype A1A1. So that means this gene A1 is the one which determines maleness, and it is dominant over A. The female therefore would be a homozygous capital A, capital A. Now on your screens you find a cross between a male and a female. A male in this case is AA1 and the female is homozygous capital A. The male would give rise to two types of gametes, whereas just one type of gametes would be produced from the homozygous female. And the progeny therefore would be the male and the female in the ratio of 1:1. Now there are no chromosomes involved. By chromosomes I mean no sex chromosomes involved, and the genes are present on different chromosomes. So in biology we call this as gonochorism. Now gonochorism would mean that there are two sexes of each individual. They are male or female, and in this type of gonochoristic vertebrates the sex determining mechanism can be classified as genotypic, as the example of which I just mentioned, which is abbreviated as GSD, or they are temperature dependent, which is called as TSD. Now this new terminology, you would appreciate viewers, is a very primitive means of sex determination in the lower forms of the animals. Now it's very common in animals even now, because we find that the two sexes in the primitive forms were differentiated just on the basis of the gametes, and it was much later in evolution than that these morphological or phenotypic differences, they actually now metamorphosed or transformed into two sexes of a species. The distinct sexes were not there in the beginning. So what was there in the primitive forms? In the primitive forms, the sex determination was actually on the autosomes, and this is what is the example for genic types. And then in the process of evolution, very gradually the genes for sex determination got localized on now specific chromosomes, and this was a evolutionary step which led to the evolution of sex chromosomes. And then we started designating these sex chromosomes as X or Y or even Z or W. Why did we give different names to them? Because they would otherwise merge or they would be confused with the autosomes, and they are usually distinguished morphologically from each other. That is the two sex chromosomes, so we call them as X and Y or Z and W, and the rest of the chromosomes would be put in just one category, which would be designated as A, that is the autosomes. So we would have the autosomes and the sex chromosomes. Let us go back to the history of how the different types of chromosomes and the whole idea crystallized. Viewers, you'll be surprised to know, if you see on the screens, we are talking about a French scientist, Pierre-Louis Monroe de Maupetit. He was a French naturalist, and imagine 100 years before Darwin, he gave the idea and he wrote in his 4-volume book, which was entitled Venus Physique, that is the earthly Venus, that somehow there was a speculation on the organismal adaptation to the environment. That means the environment played a very important role in deciding how an organism would behave sexually. And he also debated on the source of human sperms and eggs, although he did not discover the sperms or the eggs, but then he had a vague idea as to there must be some sort of propagules coming from the male and female. Imagine, viewers, this was an idea in 1745, which was 100 years before Charles Darwin. And if we were to summarize, there's Alfred Jost's explanation of genetic sex determination and the environmental sex determination. You find that the temperature, the social cues, they are very important in deciding the sex, say, of amphibians, of certain reptiles, and likewise there are certain switches which now would change the pattern to gonadal sex and the phenotypic sex, which we see now in the modern world. So that means this is called as the Jost paradigm. If we were to just summarize how this evolution of sex determination mechanisms really came about, so from top you find that in fishes we have both. By both we mean the ESD as well as GSD. So that means the environmental sex determination as well as the genetic sex determination. And gradually evolving from the fishes to the amphibians, the snakes. Now the snakes would have the genetic sex determination. Whereas again when we talk about the turtles, we have both. And ESD again comes to force in case of crocodiles. And then when we transform it, now evolved to birds, that is the aves, and the mammals, the marsupials, then it is purely the genetic sex determination. Viewers, if you were to appreciate the evolution of sex determination mechanisms in terms of time, you would see the various connotations that the whole story of fishes started, say, about 450 million years ago. They had the mechanisms rather than all the mechanisms, right? But mostly it was the environmental or the temperature control. And gradually there was an evolution, say from the amphibians to the turtles to the crocodilians, the birds, and in birds you find that there is a ZZZW type of sex determination. We will talk about this terminology a little later in our discussion. And then again there was a type of a parallel evolution or even retrogression. One went back to the temperature controls, and then later when it comes to the marsupials and the higher organisms, the higher animals, then it came out to be the XXXY type of sex determination. The discovery of the sex determining mechanism and the sex chromosomes is also very interesting, and you must appreciate, viewers, that all the genius minds have not given all the genius ideas. Sometimes their mistakes have also been to the same magnitude as their intellect. To give you an example of Aristotle, he was the one who said that sex of an offspring is determined by the heat of the male during copulation. Now, this idea is laughable today, right? And so were the other earlier naturalists. For example, Homer, he said that conception in the sheep is influenced by the direction of wind. If there is a north wind blowing, then the males would come as progeny, and if it is southward, then the females would come. A similar idea was given by Galen, who said that the semen from the left testes would make female and the right would make a male, and if there is a mixture, it would produce hermaphrodites. So they were very funny ideas. The actual scientific discovery and seriousness in sex determining mechanisms came when the microscopic techniques were employed and people actually saw the propagules. For example, the famous Dutchman Anthony van Leeuwenhoek, he observed the sperm. Ovum was observed by Van Baer, and then finally it was in 1891 where spermatogenesis in the squash bug was that is Pyrocorus, showed that there were 11 pairs of chromosomes. Now I immediately came to chromosomes. Viewers, you must remember that by this time the chromosomes had been discovered by Waldeyer, and people could now have the technique to count the chromosomes. So Henking was the first person who said that there are 11 pairs of chromosomes in meiosis. However, there is an unpaired element which moves to the poles. I will leave the discussion at this note, that this unpaired element was an X chromosome or a Y chromosome or a bar body. This we will now deliberate and come to a conclusion in our next lecture. For this lecture, I have also consulted various books which you would see later on your screens. Thank you.